As I said, thanks for the introduction. It's really an honor for me to be here and be able to share with everyone what we've been working on for the past 2.5 years, I think now. So today I'm going to talk about our project, which we titled Nucleosome Repositioning in Chronic Lymphocytic Leukemia. This project was done in collaboration with labs in the UK and in Germany. And here I've listed all people that took part and which are also currently working, taking part in writing the manuscript, which we hope to submit soon. So before I start, just a little bit of background on CLL. So chronic lymphocytic leukemia is the most common type of leukemia in adults. It is a blood cancer which affects the white blood cells, which are specifically the B lymphocytes, which are also known as B cells. These cells are not fully developed, do not work properly, and as seen from the photo here, they also look abnormally. In some CLL patients, the disease progresses slowly, while in others, it's more aggressive and requires earlier diagnostics and treatment. And because of that, prognostic biomarkers are needed. The mutation status of the IGHV gene can be used to classify CLL patients into two groups. Those that have their IGHV gene mutated, called MCLL, and those that don't have a mutation in their IGHV gene, called UCLL. And this latter group is characterized with poor prognosis. So in this project, we focused on epigenetic changes in CLL patients. So DNA variation, DNA methylation, histone modifications, and most recently, 3D genome organization has been largely studied in CLL. But little is known about the accompanying chromatin structural changes, such as nucleosome positioning and its effect on transcription factor binding and gene expression. So here I'm giving a brief overview of the experimental data that we had in our study design. So we had these nucleosome landscapes determined by MNase assisted H3 chip-seq in some MCLL, some UCLL, and some non-malignant B-cell samples. And then we went in one direction to get the coordinates of regions losing, gaining nucleosomes in each group. And then we attempted patient stratification. And then we went in another direction and we used our other data that we had, such as attack-seq, transcription factor and histone modification, chip-seq, RNA-seq, and high-seq to further complement our study. So the base for this project was basically this first plot that I'm showing here. So as you can see, there's a significantly high correlation between non-malignant B-cell H3 reads and CLL H3 reads. But we can also see that around less than 1% of nucleosomes are positioned significantly different in CLL. I'm going to give you an example. So this is the ICAM1 gene region. So the blue box represents the gene body. On the top, we have the average nucleosome occupancy profile. On the bottom, we have the average DNA methylation profiles. And if you look, we can see that there's differential nucleosome occupancy between CLL and non-malignant B cells. Then, in addition, we also observe that the nucleosomal DNA fragments are shorter in CLL patients. To disregard this difference in the nucleosome sizes, we decided to consider only fragments of size from 120 to 180 base pairs of length, which we used throughout the whole project. So by investigating the interplay between nucleosome repositioning and DNA methylation, we found that DNA methylation actually makes nucleosomes less likely to be repositioned. What I'm talking about is if we look at the graph here on the left, we can see that nucleosomes which do not really shift from their position are methylated, while nucleosomes which shift are unmethylated. Furthermore, regions undergoing changes in DNA methylation, such as differentially methylated regions identified in our samples, also undergo nucleosome repositioning. And therefore, we propose that DNA methylation stabilizes nucleosome and prevents their repositioning, just like imposing a travel ban. While on the other hand, unmethylated nucleosomes are free to travel or reposition. In our project, we formed three groups for pairwise comparison of nucleosome occupancy at 100 base pairs non-overlapping regions of the genomes to get the coordinates of regions which lose or gain nucleosomes in each condition. And today, I'll just focus on these first two groups. And here I'm showing you the DNA methylation and nucleosome occupancy profiles around just one set of regions. On the left, we have the regions losing nucleosomes in MCLL, while on the right, we have the regions gaining nucleosomes in MCLL. And we use these profiles to confirm that we're defining the right regions, the regions losing nucleosomes or regions gaining nucleosomes. So we also observed that nucleosome repositioning is associated with chromatin remodelers. So here on the left, we can see that BRG1 occupancy is high in nucleosomes which shifted, while its occupancy is low in nucleosomes which do not shift. Out of these nucleosomes which shifted, we can also see that BRG1 occupancy is again high at regions which gained nucleosomes in both serial subtypes, compared to its low occupancy observed in regions losing nucleosomes. Similarly, we also observed high enrichment of CHG1, which is another chromatin remodeler, at again regions which gain nucleosomes in CLL. Therefore, we concluded that nucleosome repositioning is an active remodeler-driven process involving CHG1 and BRG1. And then we were interested in seeing where are these regions actually located. So here on the right, I'm showing that MCLL patients show high enrichment of regions gaining nucleosomes at all genomic regions that we analyzed, while this is less pronounced in UCLL. And for now, I'm going to focus on enhancer regions. As you can see from here, they are both enriched with regions gaining nucleosomes in the two CLL subtypes. So first, we can see major changes of CLL in CLL at enhancer regions. So all enhancer regions identified in non-malignant B cells shown here on the left are characterized with both DNA methylation and nucleosome occupancy increased at the center of the region, corresponding with the fact that these enhancer regions are actually mostly inactive. While here on the right, we can see the DNA methylation and nucleosome occupancy depletion at the center of active enhancers, correlating with the fact that these enhancers are with an active status. And then, importantly, we can see that CLL patients are characterized with an increase in nucleosome occupancy at these regions compared to non-malignant B cells. And then also, we can see that nucleosome occupancy at these enhancer regions can distinguish CLL subtypes better than DNA methylation. So what else? Well, we also observed that differential nucleosome occupancy marks CLL-specific pathways. We performed pathway analysis of genes found inside the regions losing, gaining nucleosomes in the two CLL subtypes. And for example, as seen from this graph on the right, we can see that these regions which gain nucleosomes in UCL are enriched with genes involved in the B cell receptor signaling pathway, as well as many genes involved in the immune system response. So if we go back to this enrichment graph, what else can we see from here? Well, we can also see that there is high enrichment of regions gaining nucleosomes at CTCF binding sites. And most importantly, we can see that the stronger the CTCF binding site, the higher the enrichment in both CLL subtypes. So here I'll give you a brief background on CTCF. CTCF is this insulator protein that is important for the formation of chromatin loops and in the control of three-dimensional chromatin organization. And experiments done to remove CTCF binding sites have resulted in changed gene expression. Therefore, the disruption of these loops results in deregulated transcriptional activity of genes found inside the loops or nearby genes found outside. And now, by calculating nucleosome occupancy profiles at active A compartments and inactive B compartments identified in non-malignant B cells, we observed that the changes of nucleosome occupancy observed in CLL patients persist even at a larger scale of like 100,000 base pairs. We can see that nucleosome occupancy at active A compartments in non-malignant B cells is depleted, while this disappears and becomes an increase of nucleosome occupancy at the same regions in CLL patients. On the other hand, we can see that inactive B compartments are actually characterized with flat nucleosome occupancy profiles, which we'll expect because of their inactive status. And then what about CTCF? Well, the nucleosome occupancy and DNA methylation profiles around common CTCF sites, which are defined as CTCF sites present in both non-malignant B cells and CLL patients, show these nice oscillations and this depletion at the center, which allows CTCF to come and bind and perform its function. But then we also found that some CTCF sites are lost in CLL, and they're lost because of this nucleosome sitting at the center, which prevents CTCF binding. And then we also noticed this small peak here downstream of the CTCF binding site of DNA methylation. So we proposed a model. We have non-malignant B cells, and their CTCF can come and bind and do its thing. While in CLL, some CTCF sites are occupied by a nucleosome, which is further stabilized by DNA methylation mark. Well, next, we wanted to look inside the regions losing, gaining nucleosomes in each CTCF, in each CLL subtype, and look for transcription factor motifs. So the analysis revealed a number of transcription factors involved in the differentiation and functioning of white blood cells. For example, we have the immunity-related factors such as IRF1, IRF8, which are enriched in regions losing nucleosomes in MCLL. And then we have IRF3, which is enriched at regions gaining nucleosomes in MCLL and UCLL. Then we have the CLL marker, BCL6. And we also have CTCF and its interaction partner, CTCFL, which is also known as BORAS. So therefore, nucleosome repositioning in CLL can define differential transcription factor binding, which would further allow us to construct CLL subtype-specific transcription factor networks, which I'm showing here. So some of these transcription factors are common for the two CLL subtypes. I've colored them here in purple. But some others are CLL subtype specific. And an example is the EGR family of transcription factors present only in the UCLL network. So then we took not some, but all of the transcription factors and calculated nucleosome occupancy profiles around their binding sites. And we observed a clear change of nucleosome occupancy at the binding site of JUND and FOSL. And this increase of nucleosome occupancy also correlates with the low expression, decreased expression of FOSL in CLL patients determined by our RNA-seq data. So interestingly, the encoded antigen of FOSL together with JUND form the AP1 complex, which is important for both innate and acquired immunity. And then additionally, this AP1 complex has been shown to interact with chromatin remodelers. providing a possible link to the involvement of BRG1 in the active nucleosome repositioning. And then finally, we attempted patient stratification based on a number of epigenetic parameters. We performed principal component analysis to investigate this. And then what we found is that CLL can be differentiated from non-malignant B cells based on global gene expression. But CLL subtypes can't be distinguished based on that. We performed the same analysis, but using DNA methylation occupancy at promoter regions. But as you can see, distinction was not achieved again, as we see a partial overlap of the clouds here. So interestingly, based on the PCA plot here, MCLL and UCLL are not two well-defined states in terms of DNA methylation, but rather they're two continuous states with different degrees of B cell development. So the plot here on the left, which I took from the Malm et al. Paper, nicely complements the PCA on the right. As it can be seen that the DNA methylation occupancy of each sample reflects its developmental trajectory, starting with non-malignant B cells here on the left and going towards the right, passing through low, intermediate, and then high developmental progression in these samples. So then we questioned ourselves whether nucleosome occupancy would allow better patient stratification. And the answer was actually yes. So we calculated nucleosome occupancy at regions losing nucleosomes in UCLL, shown here on the left, and regions losing nucleosomes in MCLL, shown here on the right. And we performed a principal component analysis as well. And we not only can see that CLL patients are distinguished from non-malignant B cells, but we can also see that MCLL patients are clearly distinguished from UCLL. So therefore, we suggest that nucleosome occupancy at specific regions can serve as a novel biomarker for CLL patient stratification. So the take-home messages for my presentation today are that CLL subtypes display different genome organization. We also saw that some CDCF sites are swapped with a nucleosome. And then that nucleosome positioning can serve as a new biomarker, allowing diagnostics and stratification of CLL patients. And then last but not least, I want to acknowledge everyone from the Teif lab, the lab members of the COST lab, as well as all our collaborators from Germany. And thank you for attention.